So welcome to this workshop. It's going to be just a morning workshop on AI method and model for biocatalysis and synthetic biology. Most of the people are online, so you're going to have a lot of talks that are in fact online, but some are also physically present. So it's been some time that we wanted to have such a workshop. Because of course nowadays AI and synthetic biology and also in chemistry and chemical synthesis, these are hot topics. Okay, and back in 2019, we some of us organized an EMBO workshop, and we had a session on the use of machine learning in synthetic biology. That was an EMBO workshop on synthetic biology, but at that time that was not that of a hot topic. Okay. But during the COVID crisis and after the COVID crisis, we started to see more and more papers that were presenting work that made use of machine learning in synthetic biology. Okay? And similarly, in chemistry, there was really a boom of papers that were published in chemical synthesis and notably using retrosynthesis. Quite a lot of papers. At some point, one paper a week, published on this topic, making use really of artificial intelligence. So we thought like maybe it would be a good idea to have a workshop that linked together communities in computational biology and in computational chemistry that are interested by these topics using machine learning, but bring together these two communities that don't necessarily talk to one another, or don't go to the same conference, don't publish in the same journal, because probably they are facing the same issues using these machine learning tools. And so we went on and did this workshop with the support from the one large program called France 2030, and in particular the PPR bioproduction project, quite a substantial project, who is financing this workshop, along with the biosynthesis and the IRN Synthesis Bio, which is a CNRS organization who is also providing funding. And actually, there's a conference, a follow-up conference that just started this afternoon. That is the annual French synthetic biology meeting. So the program we have prepared is not really on the classical topic that you may already know that are using machine learning, but we try to see things that are, let's say, out of the box, but that could have great potential in the future. So we'll start by seeing things in chemistry, and notably there's been a lot of work, like I already said, in retrosynthesis, and but not really applied yet to biochemistry and synthetic biology. So we're going to see some talks a talk on that. Then we're going to move on looking how reservoir computing, which is another field of machine learning, has been applied to study chemical reaction network and also biochemical reaction network. And we will also see how generative models like transformers and these things that you might heard of can be used not only to generate sequences or generate molecules, but to generate networks and to study networks, and in particular metabolic networks. And we will end with a presentation by a keynote speaker on the use of machine learning for cell factories, so right in the heart of synthetic biology. So this is kind of the program. So we had also a lot of people that asked to have a oral presentation, but we could not accommodate everybody. So we also have a poster session, and we'll give you the list of posters during the break. There's a coffee break, and then during the lunch also, we provide lunch at the end of the workshop. You're also welcome to look at these posters. So having said that, I think we can start the regular session. And so our first speaker is Esther Hyde. She graduated in 2019 with a PhD in theoretical chemistry from the University of Vienna. Then she moved on in 2020 and joined the research group of GREEN at MIT, and she specialized in chemical kinetics, and she's now a research associate at TU Vienna. She worked on a variety of problems, making use of machine learning in theoretical chemistry, and including retrosynthesis and enzymatic reaction prediction, which biosynthesis. Okay, and I guess these two topics she's going to talk about during the presentation. So welcome, Esther, and I give you the floor.
Thank you so much for the nice introduction, and really welcome to my talk. I'm so happy and honored to open up the session. I'm really sorry that I cannot be here in person today, but I hope to chat with you offline. So feel free to email me anytime if you're interested in something. And what I want to talk to you today is one of my hard topics. So I'm really excited to present this to you, and it ranges all the way from machine learning with very simple models or simple neural networks to transformers and protein language models. So I'm really excited to show you also the journey that this project took. And I've already been introduced a little bit, so you already know where I did my PhD. I also was a guest researcher working on quantum mechanics and machine learning at various universities. And I then joined MIT because I wanted to work on bioretrosynthesis, specifically on cascade design for enzymes, because I do think that this really makes a change if we push for a more sustainable chemistry. And you already see at the year numbers 2020, I went there in January, I had two splendid months, and then it was 1 year of home office and all our experimental collaborations died. So the project went a little bit different than what I thought, but I hope that this talk is also a message to the young people in the room, to the PhD students and postdocs to not give up, because your work makes a difference, and I hope I can show you that in mine. And I then moved to TU Wien I'm currently a postdoc there. I was promised an assistant professorship that I should hopefully be able to start soon. And I'm interested in enzymatic reactions in various flavors, so ranging from databases to predicting the substrate ranges, computer-aided cascades, but also a more theoretical approach of how do we represent reactions in general for machine learning? How can we do retrosynthesis both for biochemical reactions and organic reactions? And some of my recent work has also focused on uncertainty quantification. I'll not talk about that today, but there's a really new cool preprint on ChemArchive that you can check out if you're interested in that. So the reason why I was interested in the whole bioretrosynthesis topic is because I wanted to be able to press a button and then say, how can I make a compound eco-friendly, selectively? That could be a pharmaceutical, a chemical, a fine chemical, a food additive, and so on. And at the same time, I wanted to be able to answer, what can I make from a compound that is maybe a waste product, that is a renewable feedstock? And the question at that time was mostly answered with organic synthesis. I mean, there are a lot of organic retrosynthesis algorithms around that will tell you how to do it. So you plug in what you want to synthesize and you give it a list of precursors and it finds a way to synthesize this. But if you go to the lab and you actually implement it, it means you run a reaction, you have some solvent, you have byproduct, you need to purify it. The reaction conditions are not suitable to each other. So you have to purify the intermediate, then run the next reaction, and so on. So overall, this multi-step reaction will be low yield because you have to do purification steps. There will be a lot of unproductive steps. It's just protection and deprotection activation, because some of those functional groups will not be compatible to each other. And you use organic solvents, energy, possibly toxic materials. So I wanted to move away from that. And of course, we all know now we can do this by enzymes. And at this time, RetroPath RL by Jean Loup came out. And this really, like, it was a game changer to me. I read this and it really made a difference to me. It was so elegant how they proposed the synthesis pathways for enzymatic reactions based on templates of varying selectivity. And with enzymatic reactions, we don't need to purify our intermediates. We don't need so many protection activation steps. And we can conduct this in aqueous solvents and ambient temperatures. And what I wanted to do during my postdoc project was to extend this to multi multi-enzyme cascades. So to be able to also control the reaction equilibria, recycle cofactors and waste. So to make something, press a button, and then the software gives you everything. We had experimental collaborators waiting on the software. They just wanted to press the button and then implement it in the lab, but we'll get to this in a second. So the project that I want to talk about and that arose from this idea is some heuristic approaches on the substrate ranges of enzymes, which give us better reaction templates, as well as similarity searches between substrates. And then I'll go into the machine learning part of this, ranging all the way from simple networks to very elaborate ones, graph convolutional neural networks and transformers. And I'll also talk a little bit about database, because one of the databases that we recently published is really making a difference in the field currently. So I hope this will also be useful to you. But before I go into this, we need to talk about two concepts of chem informatics. And since this is a very mixed audience, I wanted to make sure that all of you understand what I'm talking about, because these concepts have really shaped the projects that I did. And you will see how these two tiny details that are really simple to understand and think about have really changed what I was able to do. So the first one is a condensed graph of reaction, and it's rather simple. So you have a reaction. Here you have hydrogen reducing methanol. So it's reducing this to methanol. So it's very simple. And if we know which atom is which, we can compute something that is called a condensed graph of reaction. So what this is the graph overlay of the reactant and product graphs. So you connect everything that had either bond in the reactants or the products, for example, atom 3 and 1, and then these red edges would be doubly annotated. So there would be one annotation for the reactants, which is 0, no bond, and one annotation for the products, which is a single bond. And the second concept that we will need is a Hasse diagram. So that is something from mathematics that is used to order objects. And since that object can be anything, we can also use it to order molecules or condensed graph of reactions, because these are nothing else than pseudomolecules. So if I ran this reaction and it worked, for example, for those 3 different types of reactions, I could compute a Hasse diagram. And that really extracts the common substructures in this. It would always be the same reactive center, the same reduction, and then there could be either 2 hydrogens attached to the carbon or 1 hydrogen, 1 carbon. So you see this if you just look at this by eye. So there would be 2 hydrogens here, or 1 hydrogen, 1 carbon, and then you can extend the information on the carbon and arrive at this Hasse diagram. And we will see in a second how this really is helpful for enzymatic reactions. So let's dive into enzymatic reactions. So thinking back about RetroPath RL that I talked about a little bit in the beginning, it uses templates, so reaction rules that you extract from a database of reactions at different levels of specificity. And if you use it other projects or also organic retrosynthesis, they all extract reaction rules, or some of them, at least the ones that work on templates, extract reaction rules with some pre-specified level of specificity. So you say, okay, in my reaction rule, I include the reactive center and maybe everything that's up to 1 bond away, or maybe some functional groups that are adjacent to this. But this really doesn't work for enzymes from a chemist or biochemist perspective. So if you think about, for example, hydroxyoxyglutarate lyase, and you look up on Brenda all of the reactions it can catalyze, you see the reaction always splits a CC bond next to our hydroxyl group. So this is always the same motif. This is the reactive center, a CC bond being split next to a hydroxyl group. But if you look at the reactions more closely, you will see, especially on the product side, it's very prominent that one part of the substrate is always the same. This is the one that splits off to pyruvate. And the other side, where the hydroxyl group is, it doesn't really matter. There can be a chain of something, can even be charged. There can be something not charged and really small. It's just that 1 carbon and basically nothing else attached to it. There can even be a double bond here. So it seems like that one part of that substrate is super important to be conserved and the other one isn't. So I really wanted to come up with a way to be able to encode this in reaction templates. And if you extract some templates, so these are extracted with RD-Chiral, you will see that you get 3 different templates for those 3 reactions, even if you make templates at a very low radius, because the changes in these substrates are really at the carbon that is the reactive center. So they are very close to the reaction center. But having 3 different templates for the very same reaction that runs with the same mechanism is not really useful from a chemistry perspective. So I thought, why not transform that to condensed graph of reactions? These are very simple to compute if you know which atom is which, and then we can compute a Hasse diagram. So I implemented a Hasse diagram. This also a software that you can use for this and that you can download. It will do all of this to you. It will give you the visualizations and those trees. And what we see then is exactly what I saw there as a chemist. The reactive center is always a carbon-carbon bond next to this hydroxyl group. And then we can extend and look for common substructures. And it's always that one carbon that is the same, that is bonded to 2 hydrogens, 1 carbon, and then you extend it all the way out to this pyruvate group that then splits off. And only very late in that tree, it branches off. And what you see here is some notion of substrate promiscuity, because if you process an enzyme and the tree is very broad and very shallow, it means that this enzyme is very promiscuous. It can catalyze nearly anything if it has the correct functional groups. And if this is very elongated, it means that the enzyme is not promiscuous at all. There needs to be very large groups of atoms conserved for this to work. And we can then, for example, extract mutual specific templates. We can also relate this to the mechanisms. For example, for hydroxyoxyglutarate lyase, the mechanism is actually known. And this pyruvate part of the substrate is actually really important because it is part of the mechanism, and this carboxylate group needs to be present because it interacts with the amino acid in the active pocket. And we could see that in a data-driven way with data-driven templates at varying levels of specificity, which was really exciting to us. So this is pure chem informatics, no machine learning yet. And we then curated a large set of enzymatic assays from literature. So these are high throughput assays of a lot of different enzymes screened against a lot of different substrates. If you want to use this data, it is public on GitHub. It took me several weeks to extract this. So if you can make use of this, please do. I would be very happy. And what we can see is that if we try to classify whether an enzyme interacts with a substrate, and we do that with similarity. And similarity is what a lot of the more simple retrosynthesis algorithms actually use. It works well for some assays, and it works terribly for others. And it is especially those that are very selective, where similarity approaches fail. And that's because if an enzyme is not promiscuous, it needs very large portions of the molecule to be conserved. A good similarity score doesn't give you anything. It doesn't mean anything. You can't use it for classification because you need to look whether the parts of the substrate are conserved that actually matter. And for the rest, it doesn't really matter whether it's conserved. We could also use this for co-substrate proposal. I also wanted then to use 3D similarities in this approach, and I implemented an open source Python package to compute 3D similarities that is based on shapes and electrostatics. I don't want to talk in detail about this today, but I just want to point you towards our GitHub repo if you're interested in 3D similarities. This is open source, so it's an open source competitor to OpenEye. There's also a workshop on this that you can find on YouTube that will explain everything and how to use this. And this came in really handy for us to go beyond 2-dimensional similarity of substrates. And with that at hand, so we could make custom really good templates and we could compare the similarities pretty well. I wanted to go ahead and do some machine learning, and the group that I worked with is one of the authors of ASCOS. So if you know, this is an organic retrosynthesis platform that can design retrosynthesis pathways using organic reactions given a product which is template-based. And the way this works is you plug in a huge database. So the organic ones are usually based on USPTO and Pistachio, and then you compute all of these templates from the atom map reactions that you get in your database, and the neural network that then does the retrosynthesis for you is utterly simple. You take your product, you turn it into a 2-dimensional fingerprint. There's a neural network that is basically just one layer of neural network, and it will tell you, oh, well, this product is template 2. So this is nothing else but a multi-class classification. And as I was trying to reproduce the results for the organic retrosynthesis to get ready to also use this for enzymatic retrosynthesis, I noticed something very odd about this. This is multi-class classification. So if you think about this, for example, for image classification, and you had templates like this, so you want to classify pictures, and you have a few dogs and cats and birds, and your classes are, I don't know, snake, dog, cat, bird, then this is nowadays really easy to train. You can train this up to very high accuracy. But no one would design their classification tasks like this: reptile, dog, snake, corgi, poodle. Because what you run into then is that one thing might be classified as a dog and the other one as a corgi. But corgis are also dogs. So this should also be a true. And if the model predicts true for dog, it should get a good score. But what we instead get is false negative, because this should be positive. So these would be very difficult to train and very low in accuracy. So no one doing image classification would do that. So what we need to ensure is that our classes in multi-class classification are mutually exclusive. But are templates that we're wanting to predict here mutually exclusive? Well, I looked into this and the answer is no. So what we were using in ASCOS for a very long time is not exclusive templates. And this was the flagship software at MIT that was distributed to many customers, and no one ever thought about whether the templates are exclusive and whether it makes sense to use multi-class classification. And if you think about it, and there's, for example, a linear reaction, so this is a linear chain next to the reactive center, or a branch chain, you would get different templates. But if you apply this to a branch product, both templates apply. Both produce the correct precursor. But one of them is labeled as false in a database. So this really confuses model training. So I wanted to test how much performance is lost, because my gut feeling is that this is really major. So I hired a student to do this, and here I'm showing you results for organic reactions, but the same is also true for enzymatic reactions and other data sets. And if you plot the top-end accuracies of a retrosynthesis algorithms, so this is, you take the product, you predict the templates, you apply the templates, you get the precursors, you compare whether it's the right precursor in the top-N hits that your model outputs. And we got this curve, and this was the one that was published in the ASCOS software. And then I checked, well, is checking for the correct template even the same as checking for the correct precursor? And it was not, which already indicated that the templates are not exclusive and there's a perceived loss in performance. But there's also a true loss in performance, because we need to fix that hierarchical labeling problem. And if we go back to the simple image classification, what we would need to do is go through all of the labels and look at whether there's hierarchical relationships between them. For example, all corgis and poodles are dogs. So if something is named corgi, we need to relabel it to dog. And we can do the same for templates. This is just a little bit more tricky to get the Hasse diagrams, because we need to compute subgraph isomorphisms. But if you first group all of your templates into general templates, grouping the same reaction classes together, this is not a lot of computational work. This can be done in a few minutes for example, USPTO 50K. So this is not a big problem. And then you see, well, if those templates are the same, but just more specific, I need to use the more general one. And if you then retrain your algorithm on this, you get a really large performance increase in the top-N accuracies. And the fun thing is, this is now the same performance as all of the state-of-the-art approaches in this field, being template-based or template-free. It is just one layer of neural network, and all we did is correct for the templates. So I mean, this is a really big pitfall, and if you're working with templates, please make sure that they are mutually exclusive, because it does matter if it's a classification algorithm that you train in the end. So having fixed that, I then wanted to apply this to enzymatic reactions, because after all, I wasn't very interested in organic reactions after all. So I took RIA and MetaMDV and also Metaseq, and I plugged them into the same thing. So you run the same workflow again. You take a database, you extract the templates, you train a neural network to predict the templates given the precursors, and it utterly failed. Like if you look at the top end accuracies, the top one accuracy is below 20%. That seems not too bad for a single step, but I wanted to do multi-step. If this is 2 steps, then it's 0.2 times 0.2 is 4% accuracy. If you do 3 steps, you can't do it anymore. And what I've shown here in gray is what organic retrosynthesis on organic databases achieves. And I knew that the method worked. I knew that the neural network worked. So the problem was really with the data. And of course, those databases only have 1 reaction per database, which is a really bad predicament if you want to do machine learning on that. So I needed to curate databases first. And if you think about RIA or Metaseq, they usually have 1 reaction per enzyme. So this is not a good foundation for building machine learning models where you want the model to learn the substrate specificity. So you need several reactions per enzymes in the same way that we've already seen in that heuristic approach to substrate scopes. If you know a lot of reaction, we can hope that the model learns which part of the substrates are important and which are not. So Brinda is the way to go if you want more reactions per enzyme. And I thought that this would be a 2-week project, cleaning up Brinda and then just plug it into my algorithms, but it was not. This was like 1 painstaking year of staring at Brinda reactions and trying to clean them up, because Brinda is really messy. It is not well curated, and you have a lot of wrong reactions, and they can be wrong because the annotations are wrong, the molecules are wrong, molecules are missing. They can be wrong because it seems that some of the curators there are not chemists, and then they say, well, if, for example, an alcohol that is primary reacts to, is oxidized, it forms an aldehyde. And then I thought an alcohol that is secondary, if it is oxidized, also forms an aldehyde. But if you do this in plain text, it is not an aldehyde, it is a ketone. And if you name it aldehyde and then try to resolve that back to the molecule, back to the small string, it will add an additional carbon to the molecule, which is of course not true. It just appeared out of nowhere. So I spent a lot of work cleaning up Brinda and making this an automated framework that you can also use for other databases. So if you're interested in that, check this out. And then I went back to my retrosynthesis. And with that new database, EnzymeMap, we were actually able to achieve something that is very on par with organic retrosynthesis and that we can also use in a multi-step format, which was really exciting. And then once we had the database, so many new projects and avenues open up. For example, retroselectivity. Given the reactants, what will the retroselective products be? And that was also based on some previous work I did at MIT while the retrosynthesis wasn't working out so well. And I became the head developer of the graph convolutional neural network for molecules, which basically takes a molecule, transforms that to a graph, you encode the atoms and bonds, you exchange information in a message passing algorithm. This will give you a fingerprint, you pass it through a neural network, predict the target. And what I did was adapt that to reactions. And the idea is really simple. If you know what a condensed graph of reaction is, it is a pseudomolecule. You don't need to do any changes. You compute a pseudomolecule, you plug that into the software. You just need to change how you encode the atoms and bonds because they now have 2 labels, one for the reactants and one for the products. And this was really successful to predict activation energies, rates, yields, and reaction names. And it was also very successful to predict retroselectivity for organic reactions. So then thought, well, let's apply this to enzymatic reactions too. And it turned out that this is really successful. So this is just computing the condensed graph of reactions for enzymatic reactions. This does not even have the enzymatic sequence information. It is really just learning the retroselectivity by examples of the reactions, and we're achieving a really high accuracy there. So even in the substrate-centered view, we can encode what products are likely in a forward prediction. I then teamed up with Daniel Probst at IBM because I wanted to make this also applicable to transformer models. And they had already this work set up where they had enzymatic reactions and an encoder and decoder based on a transformer model pre-trained on organic reactions that was able to, in a forward and in a backward way, to predict enzymatic reactions and their EC numbers. And we added EnzyMap on top. Arrived at a much better model that is actually now the foundation of IBM Reaction for Chemistry. And if you go to the IBM Reaction for Chemistry website and you say predict retrosynthesis, and I'll just add this to an existing project, and you want to synthesize something, I don't know, I'm just making up something. For example, this. You can load this. This is complaining because I did a 0 instead of an O. And then we can click predict retrosynthesis mode. I'll click on interactive, but you can also do automatic. And if you scroll down and you click on enzymatic 2022, that is based on EnzyMap, which is really exciting. So this is the official IBM model. And you can then click on AI and expand this and get some suggestions on how to make this. This usually takes a second or two, and it will give you some confidence score and what EC class would probably be able to make this. It also gives you the cofactors. And if you're not happy with this, you can just expand this to another node, which is really nice. So if you're interested in that and you didn't know about that tool, check it out. It is free, but you do have to sign up. Then also the Turner Group at Manchester got really interested in our work to include this into RetroBioCut. So if you go to the RetroBioCut website and you, for example, trying to reproduce the same molecule that I did here, it's already still there, and you click on Start, you can choose the reaction rule set that you want, and you can click until this becomes EnzyMap. So you can use that database that we used, and if you now double click on a node, you can expand this and get all the reaction rules that I extracted out of EnzyMap. And you can look up the precedence for a given reaction. You can even look up the similarity to the known reactions, which is based on the Hasse diagrams that I showed you before. So you will also encounter back the Hasse diagrams. So this turned out to be really useful to the Turner Group. Very lately, just a few months ago, there was also an integrated reaction enzyme mining and evaluation platform that came up, REME. If you have not heard about this, check it out. It is really cool. You can, for example, input a reaction. I'll just use the example reaction that I did here, and I can click on search, and this will take a second. And I do hope that you can see my screen. Yes, good. And you will see here that you get the reaction, including all of the cofactors. So these are balanced, and AtomMap, because EnzyMap is balanced and validated, you see where the reaction happens. You can run similarity searches. You can use, for example, RDKit or reaction fingerprint or other fingerprints from the literature to expand your molecule. So this is really exciting. There's also one work that was just presented on ICLR a month ago that is based on EnzyMap and protein language models, which comes out also of the Regina Basile's group that I collaborated with. And this is taking from EnzyMap. It also has the protein sequences. The protein sequences encodes that with a protein language model, encodes the reaction with a graph attention model. It proposes different bond edits, and for each of the different proposals, so each of the different products, it again encodes the protein sequence. It computes from the reaction the condensed graph of reaction. So this is just my reaction chemprop that I published. So here you find that again, and combines that information, and we can then get a ranked list of products, what the method thinks that given that enzyme sequence, the product will be. And I just copy pasted that little table out of their paper, and you see that the accuracies for this method are like double the accuracies that you could get with most of the methods that we had before. So EnzymeMap really made a difference because it supplies a well-curated database to the researchers that actually do this research. So I was a little bit sad that we couldn't do the enzymatic cascade design in the end within the time frame that I had. So this project ran out last year and I submitted lots of grants and I hope to be able to pick it up again this year, but currently I'm working on something else. But it makes me really happy to see what the community made out of this, and I also hope that this is of use to you. And with that, I'm already at the end. Many thanks to Green Group and the Madsen Group, which are a big part of that. And also thank you for your attention. Feel free to ask questions or just check out my GitHub repo, find me on Google Scholar, shoot me an email and get in contact. I'm always happy to talk and also to collaborate. Thank you.
Thank you. So we have the microphone here. Anyone has question? So maybe I start. Well, yeah, online also, yeah, we can. Okay, so let me just ask one. Okay, so at some point you had you have this AST diagram and various ways of getting these things similar to templates, and basically from that you basically at some point you use multi-class classification, trying to find the templates that will apply. Okay, if I get that right.
Yes.
And you show that sometimes there's issues because there's some subgraph isomorphism problems, and one template is more general than another, and so on. But I was wondering that, and you showed examples of dogs and various animals, but there was only 4 or 5 classes. But when we are dealing with reaction templates, we have thousands of templates, okay? And so this is a multi-class classification among a huge set of classes. And so I was wondering, is that doable? And because,
It is, yes.
you know, yeah, so because personally we never did it, and I always wondered about that. When you have a huge number of classes, what kind of tools are you using for that, or just the classical tools works, or is there something to be done?
So this is a problem that I thought about very hard and that I think solved in a very nice way. So you're right. The templates that we're looking at, for example, for USPTO 50K that I was showing, this is 10K, so 10,000 templates, which is not a lot for retrosynthesis, but still a lot if you want to compute those Hasse diagrams. What you don't want to do is compute subgraph isomorphisms for all pairs in that 10,000 templates. Because then it will take a very long time. But what we can do is get 2 sets of templates. One, a very general one. For example, use RD-chiral to extract templates with radius 0. This will just be the reactive center, nothing else. And then we group them. So everything that has that same reactive center will be put in the same bucket. And then we only have 1 to 10 or sometimes 100 templates in that bucket. And computing isomorphisms for those is actually pretty fast. And then you only have corrected templates within the class. And we check that manually for a lot of reaction classes, and that is really sufficient. So what you do, and the software does it for you automatically, you really just put in your data set and you get out the reactions. You don't need to do anything. You get out the templates. It will make those general templates. It will make the specific templates. It will compute all the subgraph isomorphisms. You can use, you can make it parallel on a different number of nodes if you want to, but you don't need to. And then USPTO on my laptop takes maybe 3 minutes to process. So the subgraph isomorphisms for the 10K templates. We also did this for the larger USPTO set with 1 million reactions. Then it takes maybe 20 minutes. So not so bad. We also did this for Pistachio, which is the underlying data set for ASCOS. And then it takes maybe half an hour. So it is really not a long time, and you only need to do this once because you only compute the templates once and then you're done. So this is implemented in a very efficient way.
Okay, so thank you. Yeah. Other questions? Okay, let me get another one. Okay, so you presented a lot of things on one-step retrosynthesis, and so you have this template as diagram, and you also have a not really template-free, but based on graph neural network. And but at some point you also had result like saying, okay, I'm doing multi-step. So I was wondering what kind of because there are different strategies to do multi-step retrosynthesis, and I was wondering which strategy are you using?
So what we tried so far is, if you, for example, look into ASCOS, that is just Monte Carlo tree search. And that gives you linear pathways, which are not bad. And all of these examples that I've shown you, they usually use different versions of Monte Carlo tree search or interactive models to get to a multi-stage or multi-step pathway. And that works well for linear pathways. And for the Cascade project, I really wanted to make that also aware to cofactors. And I didn't talk about this today because it didn't really work. So I hired a postdoc, and what I wanted him to do is set up a Monte Carlo tree search that has two parts of the scoring function. One is finding a path to something that is synthesizable or that we can buy, and also recycling cofactors on the way. So this was tracking how many cofactors I made or consumed, and it was supposed to come out at a low balance or ideally zero. And that didn't work at all. We tried to use this with AI SynthFinder, so the Monte Carlo tree search that is implemented in AI SynthFinder, and it didn't work at all because there are very many hard-coded constraints in that. And if you change the loss function, it doesn't really train well anymore. So what I want to do, but this is just a plan and I didn't do it yet, is use Pareto Monte Carlo tree search. So have different objective functions that you can optimize at the same time.
Yeah, I see. Yeah, yeah, okay, yeah. Yeah, nice, yeah. So Monte Carlo tree search. Okay, so I'm not going to be the only one asking question here. One question. Yeah.
All right, I'll try to ask a question. Let's see if it makes sense. It's going to be a bit practical. So regarding multi-step, multi-enzyme cascade, things like that. So say you have a free enzyme reaction, right? And let's say your top 10 accuracy is like 0.5, right? You had numbers like this. So I just want to see if I'm getting this straight. So that means that you have 10 choice for each enzyme. So 1,000 combination, right? And you expect to have about 10%, 12.5% to be actually correct, right? That's that.
Yes. So there's two aspects to this question. One is, what does top-end accuracy measure? Because in the top-end accuracy, you get a hit when you reproduce the exact same reactant that is in your database. But of course, there are many ways to make a product. So the one that is correct in the database is not necessarily the most promising or the best one. It is just one that was recorded in literature. So we do want to have that other predictions, although they bring down the score of our top-end accuracy. In a multi-step scenario, we also need those other options. And the second aspect is, of course, if you want to find only the literature precedents with this, then if you do 5 steps, the accuracy will be super low. But it will give you a lot of suggestions of what to try. And what we envision is that an experimenter would then use this in an iterative way, similar to what I shared on the screen with, for example, IBM or RetroBioCut, to interactively expand the nodes and then select, okay, this is promising. This is something that we can do in our lab. This is something that I can also find an enzyme for, and then expand that node or highlight nodes that are most promising to them. And we don't have a fully automated way to do this yet.
Maybe a bit of a follow-up question regarding, experimental work behind the prediction. So I don't know, in your collaborators, or what do people do? Try, in fact, they do it iteratively, like you said, or they try, I don't know, like hundreds of reaction in batch and then go from that. I don't know. It's just
Yes, it is a very good question. So the collaborations that we had set out for this project, because of COVID, they never actually tested anything of that in practice for this project. So I can only tell you what they wanted to and what they planned to do. And what they planned to do was exactly in that expert fashion. They look at them and then implement what they thought was the best choice. That being said, I do have a lot of grant proposals running now where we also have experimental collaborations that would look into this in a more detailed fashion where we could do active learning loops. So have a model predict something, then have the experimenter really implement all of that, no matter how stupid they think it is, unless it is hazardous, and then get back all of the data and use it to refine a model, because it is really important to also get negative data to your model. And that is something that we don't have if we turn to Brindo or any other online database, because they only report the positives, of course.
Okay, so I think we need to stop because we're going to be too late and we need to stay on time. It's a very dense program. So I'm going to call for the next speaker. It's Delphine Dessau. Okay, she's going to be here. And she will be talking about design of symmetrical multi-component proteins using artificial intelligence.
Hello, I will present you my project on the design of symmetrical multi-component protein using a hybrid generative AI approach that was developed in collaboration with the TBI and the MIAT lab in Toulouse. So first, why using computational protein design? The experimental protein engineering techniques can only explore a tiny portion of the huge search sequence space. That's why computational protein design became a powerful tool for high-throughput pre-screening of large search space and can therefore reduce the experimental test and cost and increase the success rates. There are two main approaches of computational protein design, or CPD. The first one is based on physics, is a physics-based approach. They use 3D model structure, 3D structure or model of a target protein this approach use physics and statistics core function or fine-grained models and optimization algorithm to predict the most favorable sequences. And recently, in the lab, we introduced AI automated reasoning algorithm that could improve this physics-based protein design approach, and that I will present more in the next slide. And with the increase in data protein, with the increasing mass of data protein now available and the AI machine deep learning progress, we can now use data-driven protein design methods that can use either a 3D structure such as protein MPNN or only a sequence such as ProGPT with a coarse-grained model trained on protein data to predict the best sequences. And protein MPNN is well-known techniques that was developed in the Baker lab and use an autoregressive model to sample sequentially the amino acid of the sequence and predict the best sequence. And that I also use this in this project that I will talk also later. So for our AI automated reasoning method using physics-based design, we use a 3D protein structure template and a design space definition to compute a score matrix with pairwise decomposable scoring function. This approach can is using an automated reasoning algorithm, the cost function network, to explore the combinatorial search space and find the sequence that will optimize the score to its global optimum. This method is accelerating the design process with several orders of magnitude and outperforms the previous computational method in terms of computational accuracy and efficiency. It can also generate libraries of near-optimal solutions with the control and guaranteed diversity. And while the previous method, most of the previous methods are only using a single state of the target protein, this method can integrate several different states and then predict sequences that are compatible with several molecular or conformational states crucial for the protein function. But a lot of the applications that we need this new protein need a high specificity, either in the binding of a ligand for enzyme or protein partner in protein complexes. And in this case, we need also to consider the unfavorable states that we call also negative state. That gives a B-objective optimization problem with favorable and unfavorable states. And we need to find a sequence that will optimize the favorable states, the score on the favorable state, while disfavoring the negative states. It is extremely challenging with a physics-based approach, since the physics tends to favor the stable conformations, while the design objective wants to destabilize the negative states. To bypass this drawback, we developed a decomposable deep learn scoring function called EFI that learn on the structure in the PDB and could be combined with the automated reasoning algorithm of the tool that I presented before, since it's also a decomposable score function. So it is an hybrid IA integrative generative approach because it is using two different types of AI methods, the deep learning and the automated reasoning approaches. So I use this approach to design a symmetrical multi-component protein. So this, the protein that was chosen is involved in objects that we find in bacteria that are called bacterial microcompartments and that have a role of a bioreactor because it sequesters the different metabolic pathway to concentrate the molecule involved in the pathway, but also to avoid possibly toxic release of intermediates. And the shell of this object is composed of hexamers and trimers for the facets and pentamers for the vertices. The hexamers are the major component of the shell, and we chose a protein called RMM that can be produced in Escherichia coli and self-assembled in Homo hexamer and was crystallized. So you see here the hexamer with 93 residue by monomer. And our design objective was to design two different proteins from this template that will assemble in an heterohexamer, but without forming the two Homo hexamer. So we use this B-objective optimization problem that I presented before. So for answering this problem, we use our hybrid AI generative approach, the EFI-Custozyme approach, and also the protein MPNN approach developed by the Baker Lab to compare the efficiency to answer this negative design problem. So first, we define the interface, the designable residue at the interface between the monomers, and we applied different, we performed different design by varying the weights applied on the positive and negative state, resulting in around 200 design solution by approaches. And each solution is composed of two sequences, the A and B sequence. So we also constructed the conformation for each state for each solution, and we minimize this conformation with a fine-grained score function, a Rosetta fine-grained score function that can alter the conformation and also can alter the conformation. And we scored the conformation before and after minimization with both EFI and protein MPNN score function. So in order to in order to determine if the solution could answer our design problem, we calculated, you don't see the formula because there is people before, but we computed the difference between the score of the positive state and the score of each negative state that we call delta A and delta B. So lower are as the score difference, better is the answer to our design problem because the AB state is more stable than the negative state. And we represented this result in this kind of plot with each dot corresponding to one solution. And it's a delta B in function of delta A. And in order to better determine the capability of each design method to answer to the problem, we also introduce a gamma parameter. You just need to understand that the higher the gamma parameter, the better the method to answer the design problem. So first, I will show you progressively the result. So first, on the left, it's a design performed with EFI, and on the right, the one performed with Protein MPNN. We scored the score here is the score differences calculated with the Protein MPNN scoring before minimization. As you can see here, and also with the gamma here, we have more the score differences with the EFI design are lower than the one for the Protein MPNN, even if it's Protein MPNN that is scoring here. And when we are scoring with EFI, we have the same tendency, but it's even worse for the result of Protein MPNN design that are more with the differences more in the positive area of the graph. That shows that Protein MPNN struggles to recover the low score sequences with which answer our design negative problem. And after minimization, we have more scatter plots that comes from the big distortion of the conformation that we can have that obviously are influencing the scores. But we can still see that we have more negative differences for EFI design than for Protein MPNN. And this is still also reproduced with Protein MPNN. With Protein MPNN design, and but we seem to have stronger discrimination with EFI than with Protein MPNN. Sorry. And just quickly for the experimental characterization, we chose the best design to perform experimental characterization with GFP tripartite technology that is developed in the lab. So we are expressing both partner with one part of the GFP, so the A and B, and only if we have the heteromeric interaction, we have fluorescence emission. So after production in expression in Escherichia coli, we measured the fluorescence, and as you can see here, we have more fluorescence for the EFI designs than for the Protein MPNN. And the best design is one with EFI and has as much fluorescence than the wild type control. So EFI also experimentally seems to be better than Protein MPNN at predicting sequence of interacting monomers. To conclude, we used an hybrid AI generative approach that combines deep learning, coarse-grained score function, and multi-state automated reasoning design tool to perform negative design strategies that succeeded in engineering mutant bacterial microcompartment proteins, and also in silico as well as experimentally, our EFI approach seems to have better results than the Protein MPNN approach. And I would like to thank all the collaborators in both labs and you for your attention.
Okay, so we just have time for one quick question. Yes.
Thank you. I had a question about like, so your two monomers, like, so you start from a scaffold which is already existing and you use your program to actually generate mutations. So how far away are they? And have you tried to actually generate completely new sequences, entirely new sequences, you know, based on the structure? So can you explain?
So the first part of the question was?
How far, so your two monomers actually,
Yeah.
they're based on a natural scaffold, right?
Yeah.
And then you introduce specific mutation to favor hetero metamerization. So that's right? It's a natural scaffold that you mutate.
Yeah.
And how far away are they? And the second question is like, have you tried starting from the structure, de novo generating actually totally entirely new sequence?
Okay, so I will start with the last part of the question. So yeah, we try. We didn't try to until the experimental part, the de novo design with the scaffold still, but we tried it. We have we have a result in a computational time quite reasonable, but yeah, it was maybe too challenging for the experimental part, so we decided to limit to the interface. Also, the more exposed residues are sometimes too, for the analysis, maybe a little too similar. I mean, with the EFI approach, we are still improving it with the new information. And since the fold of both monomers are really, in the beginning, are identical, we are thinking that just the interface is enough for the test. We also did, I didn't present for the other result, but we also did a little more extended region with around 10 residue more than the 29 that we did here. And we had the same kind of tendency of results. And for the first part, when you say how far, you mean diversity of sequence? Yeah, the diversity of sequences are around, I think, okay, for, I did it, I calculated it. And it's similar between protein MPNN and EFI. Maybe there are a little more diversity with EFI, but I am not completely sure if it's EFI. But it's in the same order of magnitude of diversity between sequences, between both approaches. Is that the question?
Okay, thank you.
Thank you.
Next speaker will be online. Is Professor Willem Uck. Willem is a chemist with a PhD from Twente University in the Netherlands. So he has been a lecturer, then a reader, then a full professor at Cambridge University in the UK. And he's been spent there more than 10 years. So he's now a professor at Radboud University, also in the Netherlands. He's got many prizes and honors, and among them, he's a recipient of an ERC Advanced Grant. And so he's been interested toward understanding how chemistry can create biology, so the link between chemistry and biology. And in particular, he has worked on prebiotic, the foremost prebiotic network, and also enzymatic reaction network. And he has been using this concept of reservoir computing. And I suppose his presentation is going to be around that reaction network and the use of reservoir to understand their capacity. So thank you. And I'm going to leave you the screen.
Yeah, I hope you can all hear me.
Yes, we can hear you fine.
Let's get started. Right, yes, you already mentioned I'm very much interested in complex reaction networks, and maybe it will be a little bit of a side sort of lecture to many of your expertises in the audience. But yeah, when I look at this, I'm just simply amazed that this is apparently what you need to make E. coli do what E. coli does. And so that is in a way a continuous inspiration to see, can we do something like this? Can we build something as complex as E. coli? And what do we need to do to understand these kind of systems? So in the past couple of years, we have really been studying reaction networks, whether these are composed of very small molecules, and I will mention them a little bit later on, where you really start with just formaldehyde, nothing much else, and you create a lot of different sugars, or we design our own reaction networks and enzymatic reaction networks, and we lack the tools that Esther was just explaining, and Professor Miskovic will talk about later on. So we work with very small reaction networks and see if we can make them sort of exhibit interesting properties. And we are also very much interested in cellular reaction networks. Can we develop tools to actually observe the dynamics of, say, signaling networks in living systems? So this is very broad. I'm not going to discuss all of those topics today. I wanted to talk briefly about enzymatic reaction networks and then also introduce the concept of reservoir computing to you. So we have been working a lot with sort of enzymatic reaction networks, and this is not a new reaction network. We actually just took it from nature. This is the sort of nucleotide salvage pathway. So it's a small-ish number of proteins converting essentially an activated sugar, coupling it to the nuclear bases, making the nucleotide triphosphates. So nothing too complex, but what we really wanted to know is sort of, can we predict the dynamics of these kind of networks? Can we extract the reaction rate constants? And actually that sounds very simple, but it's actually rather difficult, because if you go to Brenda, you will find that every one of these enzymes is described by many different mechanisms. There might be hidden interactions. So if you add everything together, it might actually become quite difficult. And so what we did is actually we immobilized all the enzymes. We put them in a continuous stirred tank reactor. So it's just a small reactor with a stirring bar in there. All the enzymes are immobilized on hydrogel beads. And then you have your inputs are your substrates and your cofactors. And what comes out is still the inputs, of course, and all the intermediates and the final products, which we then observe using HPLC. So we take hundreds of HPLC spectra while we are constantly changing the inputs. And why are we constantly changing the inputs is because Bob built a little algorithm that said, well, if we want to, in one sort of big system, extract the kinetic information, then of course you could imagine that if you increase the concentration of one of these starting materials, you will automatically increase the rate of product formation, but you cannot really say, is this due to any particular reaction rate changing, or is this sort of coupled by something going up and something going down? So you want to be sort of maximally informative in such a way that you sort of try and decouple the balances, the collinearities in your system. So this is the experimental setup. The top slide that you see here is your algorithm that says, well, you have to change the total flow rate, you have to change the input concentrations of every one of your input compounds, and then you observe the formation of product, you observe the disappearance of the intermediates or inputs, and then you try and fit your kinetic model to every one of these different traces that you get. And because you have removed the collinearities as much as you possibly can, you probably can sort of, in one long experiment of say 15 hours of pulsing and obtaining samples, you actually sort of get a fairly good match to the underlying dynamics of the system. Even if it's not the perfect match, it allows you now to control or predict the dynamics of the system. So I can, for example, ask the question, what do I need to do to get an output ratio of 3 to 1 to 1 of my ATP, UTP, GTP, or 1 to 2 to 3, and a certain minimum conversion efficiency. And then you see all these particular solutions to that particular question. And you can then test a couple of them and indeed observe that, yes, indeed, we get a good prediction of the output, except when the overall concentration becomes very small, and that's because our HPLC is not very sensitive in the very low concentration regime, so then it breaks down. So essentially we can play around with input concentrations to have a complex network have a certain output concentration. And I won't go into details on the actual model that we have. It's in the Nature Communications paper. What we recently did is we said, well, the problem with the HPLC method is that it only gives you 8 different observables or 15 different observables, and it's a very slow method. So what we did in this case is we took the glycolysis pathway, we immobilized 13 enzymes on hydrogel beads, we put them in a flow reactor, and then we immediately inject the outlet of the reactor into a mass spectrometer. So now you get a data point every half a second. You can follow this for a very long time. And I can see all the different observables in there because the mass spec picks up all the different small molecules that are present in the network. And if you use isotopic labeling, then actually you can quantify as well how much of each of these compounds you have. So you really get a lot of information. And so what you see here is the output of every one of the some of the species. And you can see here our model now becomes very strongly predictive. Because you have so much more observables in your system and allowing you to really sort of extract the kinetic information very nicely. So this I could explain in much more detail, but I didn't want to do that because there was actually an expert after the break who is actually much better at this than we are. And so I wanted to take you in a slightly different direction. Because if we have these reaction networks, then can we also go back and say, in biology, what are these networks actually doing? And to a large extent, they're actually processing information from the environment. Where's the food? Where are maybe dangerous chemicals? Where do I need to move to? Where is the light? All of these sort of environmental parameters need to be sensed in a continuous fashion, and the cell needs to make decisions on what to do with that information. So in a way, you could say these large reaction networks are like computers, but very different computers from the way we normally work. And there have been a lot of efforts on DNA-based computing, but that's very often still inspired by, say, Boolean logic and sort of the computing language that we normally also use for electronic computers. And we didn't really want to do that. We wanted to sort of use these sort of molecular networks as a different type of computing. And so what we would like to do is say, well, can we actually treat these reaction networks as some kind of reservoir, as a black box in a way, where you say, well, all the compounds in there, all the reactions or enzymes are coupled in such a way that there is a huge nonlinearity. If I change input concentrations, which I just showed you, we have been doing a lot in the enzymatic reaction networks, I sort of pump information into the reservoir and I generate a readout layer, which I've also shown you, in the form of many different intermediates and products that come out. And so in reservoir computing, this is a computational framework that is very good for processing temporal or sequential data. And it essentially requires that this is a very complex sort of network that generates a high dimensional output. There are many nonlinear interactions. It should have a fading memory. And so we have that because we have a flow reactor. And there should be separation of input signals. Well, that was the whole idea of maximally informative data sets, that essentially the inputs that you give propagate through the network in a maximal sort of non-collinear fashion. So this works normally in silico, has never been demonstrated in a chemical reaction network. And so that's what we set out to do first. And so the reaction network that we use is this sort of network that I showed you on the previous slide. You start with formaldehyde, you add dihydroxyacetone, calcium chloride, and sodium hydroxide. So you run this in the presence of calcium hydroxide at pH 10, 12, 14, if you wish. What happens within minutes, there are so many reactive groups appearing because you create enolates, and then you do aldol reactions, retroaldol reactions, proton transfer, and every one of the new molecules that you generate can form any of the other reactions that were used to create them. So you get a very, very complex reaction network. We studied this before, so we have actually a good understanding of what happens. But in this case, it doesn't really matter what exactly happens in your reaction network. What we do is we We again have the flow reactor. We directly inject this into a mass spectrometer, and we follow about 100 different ion traces in the mass spectrometer. So there's a lot of data coming out. So for 24 hours, because we can run this for a long time, you continuously spray into the mass spectrometer, you follow 100 traces, you get a data point every half a second. So you're looking at 15 to 20 million data points. It's a large amount of data coming at you. So now what are you going to try and do? The way it looks like is this is our flow reactor. This is also the one we used previously for the enzymes on beets. Looks a bit like this. Your inlet, outlet, multiple inlets. They go into the mass spectrometer. The mass spectrometer is sitting over there. Here are the pumps, the reactor, and your a little bit of a dilution line to make sure that you don't spray everything into the mass spec. So what we're going to try and do is have a reservoir computer that can classify the environment, understand changes in the environment, maybe even predict changes in chaotic systems. And so the concept is as follows. So what you have is you have a certain input. The input over time changes, and that can be formaldehyde concentration, dihydroxyacetone concentration, sodium hydroxide concentration, anything that goes into the reactor. And online, you see all these 100 traces of ions. They continuously change, of course, because you're changing your input, and you keep track of this over time. So over time, you record what is changing. Now, what you would like to say is, well, my reservoir computer has to compute a certain function. And this function can be anything. It can be just a normal function, whatever you put in. You have a u squared, can be a differential equation, can be anything you want. But it does mean that at any point in time, the value of u needs to be transformed into the output of whatever function you're interested in. So that must mean that if I look at any point in time, this list of compounds that I get out needs to be summed up and correspond to this value over here. Well, that doesn't work, of course. So I need to multiply it by a set of weights that when I multiply every value of x by the set of weight w, it actually gives me this value, which it will also not be the case. So I need to have a training step. I need to collect data for a certain amount of time and then minimize this function here, which is a linear regression problem, where I say, find me one set of weights that would satisfy for all time points, generating a series of distributions of ion traces that sort of, in the end, will match as closely as possible the outcome of this function. So this sounds very complex. Actually, that's not the case, because all the complex bit happens in your reservoir. All the nonlinear transformations happen here. This is just a linear linear regression. So how are we going to do this? Well, you could, for example, start with a static problem. You say, check for many different combinations of formaldehyde and sodium hydroxide. I do an experiment. I get a certain output. And then say, well, suppose now I have as my target that all of these should be a 1, all of these should be a 0, a cat and a dog. So if they all add up to a 1, I need to find a set of weights that convert all of these compositions into a 1 and all of these compositions into a 0. So that's possible, actually. It works really well. So I could say, take my 100 experiments, classify them as an AND gate, or an OR gate, or an XOR gate, or checkers, circles, sign. I have one set of data, and I can now reclassify based on that same set of data. I simply find another set of weights. And what you see here is that for even this simple problem where you would say, I'm not even making use of the time component, my Foremost reservoir is competitive or outcompetes machine learning classifiers. So if I compare this same data set and I use in silico, an echo-state network, a multi-layer perceptron, Gaussian process classifier, support vector classifier, as you can see, I'm in the same ballpark with my Foremost reservoir. Sometimes I'm actually a lot better. Sometimes for some difficult patterns, support vector classifiers is a little bit better. Multi-layer perceptron is sometimes a bit better, sometimes a bit worse. It's at least the same ballpark. Then you say, well, can I do something more complex? Can I take a model, say a metabolic model of E. coli containing 87 substrates, 92 different reactions, set up a set of ODEs, which you can download, and predict if I have a continuously changing input into this model of carbon metabolism, I can calculate all these 87 different substrates over time. Now I can do a similar input into my reservoir, get a certain output and train, and see if I can match them. And so that's what we're doing here. So this is the in silico model. There's also dihydroxyacetone. I can stimulate an input. I can simulate the output of all the different intermediates. Now what you do is I take dihydroxyacetone in my reactor. I have a certain change in input, and I get a certain change in output. If you then say, well, now let's train this, I can train it on, say, the pyruvate trace. The red one is the in silico prediction. My yellow one is the training that I did to find the right set of weights. And then you can see for the next 1.5 hours or so, my reservoir can predict the fluctuations in pyruvate reasonably well. Same for 3PG, AMP, also very good. I can, in fact, do this for all the different intermediates in my glycolysis or metabolism network, whether it's micromolar concentration or millimolar concentration. You can see here, it actually maps on very, very well. So my reservoir computer is able to pick up the dynamics in this very large complex network, essentially learn those dynamics, and now keep predicting them over time. So this works really well for this small molecule reaction network. It works really well because the Formers reaction is so highly nonlinear. And the question that we had is, we thought, would this also work so well if you actually take an enzymatic reaction network? Well, there are no enzymatic reaction networks with the same level of complexity, so we had to set out from scratch. So the Formers reaction essentially forms it by itself. You just add formaldehyde and dihydroxyacetone. We don't have to do anything. For enzymes, we had to design this. So what did we do is we took a number of different enzymes that are all proteases. So you take trypsin, chymotrypsin, allostate, thrombin, termolysin, and an endopeptidase, and phosphatase, which is not a protease, but you could then use substrates that are phosphorylated, you dephosphorylated, and then they become substrates for any of these other enzymes. And then you take peptides. They can be cleaved by more than one of the different proteases that we have, some of them even by almost all of them. So you would say, well, that will just become a mess. But the idea would be that you say, well, if I have all these peptides in here, they will give you a nonlinear effect, because we chemists, biochemists, are used to go to the Brenda database and you say, oh, okay, if I have all these different enzymes, they have all these different substrates, I set up a ODE, and I can predict the conversion over time. That doesn't actually work that way, because this assumes that there's nothing else in your reaction mixture. If you have multiple substrates competing for the same enzyme, then actually it could well be that some of these substrates are really slow, and so therefore they occupy the active site for a really long time, which means that the enzyme is no longer available for reaction with your substrate. So you get a certain sort of well, inhibition reaction, or a certain competition for catalytic resources. And that is a highly nonlinear process, because it, of course, depends on the concentration of the slow substrate compared to the fast substrate over time. So very difficult to predict. The other thing is that after I cleave some of the peptides, so suppose it's cleaved by chymotrypsin before it can be cleaved by trypsin, then actually maybe the substrate, the peptide fragment, is now no longer a very good substrate for the trypsin, because trypsin is an endopeptidase, and now the sort of actual cleavage site is no longer sort of an endo fashion, or the neighboring amino acid sequence is no longer optimal. So that also means that the sequence of sort of cleavage actually determines the reactivity of the substrate to the enzyme. So with that in mind, we thought, well, this could well give you reservoir capabilities. So this is the different peptides that we synthesize. And then just to check, you could say, well, I can now, so what we again now do is we have our reactor, all the enzymes are loaded on beads. So whatever goes sort of, so the beads are the hardware of this reactor in a way, of this computer, the peptides are the software. So in the Formers reaction, everything flushes through and comes out of the other side. and it keeps reacting. In this case, actually, it's fixed afterwards. So you flush it in, you flush it out, and then you just have a mixture of peptides. So if you then take this mixture of peptides and you say, well, first of all, could I take every individual peptide and flush it through the reactor that contains all the different enzymes? And if you do that, and you take an HPLC, then for each of the different peptides, you will see a particular cleavage pattern because it will be cleaved by multiple of the enzymes. Some of them will cleave better than others. Some of them are really slow, so they will have a larger peak of the sort of individual peptide. Doesn't really matter what we're actually looking at, the pattern is very important. So if you say, well, let's take all of these individual traces and then compare it to an experiment where I took the same concentration of all the peptides, but I mixed them and then flushed them through the reactor all together at the same time. So same reaction time, but now all together. If you compare them, then you can see what is happening. Due to the effects that I just explained, I don't actually have an overlap between something that should be exactly the same. This was all the individual peptides going through the reactor. This was all the same peptides, same concentration going through the reactor at the same time. Gives you a different result. So this is very encouraging because this does mean something very nonlinear is happening in your reactor. So does this work? So now we can say, okay, so we have our reservoir computer. We have peptides as an input buffers. We can think of all kinds of different things. I'll get to that in a second. What comes out, we again can read by mass spectrometry, and we get a list of peaks, and we have to find a set of weights. What you see here is that I'm now no longer interested so much in actual hardcore computing because enzymes are slow. We don't have as many enzymes as reactions that we had in the foremost reaction. So my nonlinearity and high dimensionality of the space is going to be smaller. So as a computer, it might be too slow. But enzymes are sensitive to any change in the environment. So if I change the temperature, if I change the pH, all my enzymes will change reactivity, but not in the same way. So I create nonlinearity by changing temperature or pH. And I can also think later on, I haven't done that yet, by adding photoinhibitors, and then I could also use light. So where do we want to go to? I want to make this into a sensor that doesn't have any particular sensing element for pH, temperature, light, maybe ions, but because any change in the environment will change the relative rates of the different reactions and therefore lead to a nonlinear change in the output, I think my enzyme reservoir can become an extremely versatile sensor that can sense its environment. And so how do we go about this? Well, first of all, we checked again with the peptides. So I can change peptide concentration by taking half the peptide in syringe 1, the other half in syringe 2, syringe 1, syringe 2, different mix of peptides in there. I can combine them into lots of different combinations. I, again, get a list of peaks out. I say, well, some of them have to be a 1, some of them have to be a 0, and then I train to my target function. So I can do an AND gate, OR gate, XOR gate, circle. It kind of works. So I get decent scores, not as high as the foremost, not so competitive compared to machine learning algorithms, but it's decent. So what I show you here is at least it is capable of nonlinear classification. I don't really want to do sort of time series at the moment because it's about 10 times slower, this enzyme reaction, so it takes too much of a a collection of data, but I wanted to go to what senses the environment. So suppose now we still take the peptide concentration, but at the other one I change pH or I change temperature. What you see here is that indeed I can now do nonlinear classification by essentially changing pH or changing temperature. And again, the scores are not terrific, but they are at the level of many of the machine learning algorithms that you have. So this is the very first demonstration. I hope that I could have shown you more, but we are still working on more extensive sensing applications. But I think it's sort of a demonstration that this network can feel the temperature, taste the pH without having specific receptors for this. Of course, we did test the pH dependence of every individual enzyme, and then indeed you see they all have their maximum somewhere else. And so this is the underlying reason why this works. So where do we want to go and take this in the future? Well, at the moment we are collecting data, processing the data, then we and simply do the classification offline. You could say, well, I can make a system where I sort of have a certain syringe setting, I measure the output, I know I've trained it before, and I can then sort of automatically sort of update my syringe input so I can make this into a self-driving model. I could also make multiple reactors in parallel or in series, so you make deep reservoirs, improve the computational properties. In the enzyme reaction network, actually, we can do more than just tuning the or finding the set of weights on the output values. We could also start changing the hyperparameters of the network itself. And the hyperparameters are the, for example, concentration of every enzyme, the reactivity of each enzyme towards a certain substrate, the concentration of every peptide, et cetera, et cetera. So we can change many more things there, and then optimize it for a particular type of computation. And then if you combine all of these, could we make a system that automatically moves towards a sort of source of heat? So can we make this into the sort of chemical brain of a soft robot? Or can we make sensors for artificial skin? Or can we ultimately, yes, indeed, make a computer that is sort of running on molecules rather than on electrons? These are some far-fetched ideas. We don't really know yet how to do this. It's more for inspiration of discussion. So with that, I want to thank the people who have been doing the work. Bob, Quentin, and Jeroen have been working on the maximally informative data sets, foremost reservoir computing. This paper will come out soon. Mathieu has been the driving force there together with Thijs and Will, Quentin, and the enzyme reservoir computing has been taken up more recently by especially Solvik, Anna, and Rihanna, who are sort of pushing this further. And with that, I would like to thank you for your attention, and I'm happy to answer questions.
Yeah, thank you. Okay, so there's one question over there.
Hello, it's a really inspiring talk. I was just curious, in your first example, foremost reservoir computing, you were trying to model a dynamic system. Am I wrong to assume that in order to do it properly, in your physical reservoir, you should have all type of reactions that cover multiple dynamic modes? Because if you miss certain super slow dynamic modes or super fast dynamic modes, then you wouldn't be able to try to model this type of system. So in general, how would you control it in the general case? Because you put forward the idea of chemical brain, but then you would have a wide range of systems with different dynamics.
This is an excellent point, which I should have stressed myself. This is exactly why the foremost reaction reservoir can actually learn the dynamics of the enzymatic reaction networks. So you're totally right. So if you want to have a good reservoir computer that can predict the dynamics of dynamic systems, then it needs to have some of this inherent dynamics already in there. And that's why actually electronic reservoir computers are so bad at doing this, because the electronic systems are typically way too fast. So the dynamics of electrons in a computer is way too fast. So actually, if you have a lot of systems around you, they are driven by chemical reactions. Everything in biology is driven by chemical reactions. And so therefore, in Chemico reservoir computer has this intrinsic dynamics already. I totally agree with you. It might well be that you still don't cover all of the dynamics that you would find in an enzymatic reaction. In our case, we don't really find this because we sort of see very good mapping for sort of intermediates at many different concentrations, which makes it believe, at least sort of at the moment, that we probably captured the dynamics associated with those reactions. But for example, the very fast reactions in the foremost reaction, like proton transfer, probably do not contribute to the computing capabilities of the foremost. So if you think further on how to improve a chemical reservoir computer, you would like to expand the diversity of the reaction such that you capture more and more fast and slow dynamics in your network. So I totally agree. This is a very important design element of your chemical reservoir computer.
I may follow up with another question. Just I failed to understand in the case of classification, because this is still a, let me say, dynamic system that evolves. At what is the point where you are in fact collecting your output? Because
yeah, so
classification is static in general.
Yeah, so in that case, what you do is you let the system go to a certain steady state, and you only collect the steady state information. So you say, let's say every 5 minutes, I change the input, I wait 10 minutes, it's at the steady state, I take that information, and then that is what I record on the sort of XY sort of axis. I have those kind of compositions. So totally true. So for a steady state, this is a bit overkill because I don't actually make use of the inherent dynamics in my network. I just go to a steady state. So I just change the input, wait for a certain amount of time, have a certain output, and use that to classify. It's a slow process. So if you want to really do image recognition, this would take forever. So you wouldn't really do that. So it's really much more suitable for dynamic systems than for steady state. The reason why we do it is because everybody who does reservoir computing always shows that you can do nonlinear classification of images. So if you don't do it, then the reviewer will ask for it.
Okay, so I would add some questions, but they told me we need one question. Yeah, I was wondering how you wanted to follow up. Okay, go ahead.
If you don't mind, if we come back to your first example where you had this, where you were trying to estimate the kinetic parameters of a network.
Yeah.
So there, if I well understood, that was done in vitro, isn't it?
Yeah.
So for this, I mean, I'm also very interested in this question. Have you really observed that just by measuring a few, not all of the concentrations that you have, let me say, observability, that you have enough information to estimate your, because in your case, you have all the measurements. I'm wondering, did you try to estimate the parameters without using certain measurements? Because some of them might be redundant.
True. Actually, they're designed not to be redundant, but what you find in the Nature Comms paper that actually with this kind of level of observation that we have in the HPLC, we can really quantify about 8 species. It's not enough to actually make a model that is where your error bars on the rate constants and equilibrium binding constants are sufficiently small. It is good enough to predict the overall dynamics of the network, but you don't get a perfect fit. And so what we find when we go to the sort of quantitative mass spec, you can see more species in your network and your predictability becomes better. Actually, we're now working on yet another expansion where we no longer immobilize the enzymes, and we can also pulse the concentration of the enzymes in the input, and then it becomes even better. So essentially, if you want to avoid doing multiple loops with multiple changes of input concentrations, then the more observability and the more things you can change in your input parameters will always help.
Thank you very much.
Okay, so we are going to start again. We are going to start the second session of this workshop. Thank you again, everybody, for your participation. I am pleased to introduce Lublisa Miskovic. He holds a PhD degree in automatic control from the Swiss Federal Institute of Technology, EPFL. After that, he did a postdoc at the Université Catholique de Louvain and another one at the EPFL, where he is now since 2010 as a research scientist. His research interests include system biology, metabolic engineering, synthetic biology, data-driven control design, system identification, stochastic processes, and estimation theory, and the use of AI and machine learning in this field. Today, he's going to talk about generative models for metabolic networks.
Thank you, Philip. So I would like first to thank to the organizers for inviting me to present our work. As Philip was hinting, in fact, this work is probably output of more than 10 years of our research in developing dynamic models of metabolism. And though we explicitly work on really dynamic models of metabolism, many of these methods can be applied for any biochemical reaction networks where you would like to observe a temporal evolution of the states. And when I am talking about dynamic models of metabolism, we are in fact inferring to systems of ordinary differential equations. And why we would like to have these models, it is for biotechnological applications. If we would like to drive our cell factory or cellular system to certain optimal performance, we would like to know which parts of which genes in the network should be upregulated or downregulated. So in that respect, these models, they have to be mechanistic, not models of, let me say, bird view, where you just see at outputs and inputs and you try to identify some parameters so that you reproduce certain behavior. In fact, you need to know much more, because if you would like to assist to experimentalists in engineering certain system, you would need to know which specifically parts of the metabolism should be modified. What are the metabolic shifts that should be applied? And in the case of, you can use these models also in health applications, where you can either try to analyze the fate of certain drug metabolism, or you can also try to find some patterns in some diseases. For example, in cancer, it is well observed that there is metabolic shifts. And by the way, we are now working on models like this to see what is happening there. So why we are really into this ODS? Because the main issue is, first of all, you can see the small changes in metabolism as they progress, because most of the, for example, in human, most of diseases are not just a point mutation and you just lose functionality of a certain gene. These are much subtler, much finer changes. So, and these type of changes you can only capture with this type of models, because you probably have heard about genome scale models, which are in fact steady state models. In these models, you cannot investigate, in fact, regulatory circuits. In contrast with the dynamic models, this is possible. This is one, to say, advantage of these models with respect to classical genome scale models. And another advantage is that these models, they combine together multiple things in the single mathematical expression, because you have metabolite concentration, reaction rate, total amount of enzymes in one single expression, which allows you to integrate multiple omics data simultaneously. So you can integrate transcriptomics, proteomics, fluxomics, and metabolomics data in a platform such as this, and try to reconcile all these data together. Now, though these models, they are offering us a lot of possibilities, the problem is that for many of parameters that you need for these models, the actual values are not measured or are unknown. And even if they are measured, most of the time they are measured in vitro. And another problem is that different labs measure the same parameter with we have observed in some cases when you check values from Brenda, that the difference in parameters is 2 or 3 orders of magnitude, which, I mean, at least from our point of view, it is not very useful information, because we don't know what to trust. So our approach there is to try to really try to integrate all available data that are measured, metabolomics, fluxomics, transcriptomics, proteomics, if possible, and then based on responses of our model, try to reconcile all these parameters together. And whomever was working here, the main problem is how to identify these parameters. And essentially, there are two approaches. The idea is that you form it in a mathematical way where you get kind of some non-linear dependency of your desired behavior with respect to your parameters. You form this mathematical model, and one approach to find your parameters is you try to do parameter estimation. This is well known in systems and control theory. You find some initial point, and you try to minimize certain goodness of it, and you get your responses of the system. Now, this method is applied by many groups, and it works well in many cases. However, the problem is that this is a non-linear optimization problem, so you might stuck in some local minima. And second thing is that sometimes certain parameters are occasionally meaningless. And as I said in the very beginning, it is very important if you would like to propose to experimentalists that he is supposed to upregulate certain gene, that you have certain certainty about your predictions. Another approach that we are advocating normally it is to do instead Monte Carlo sampling. So rather than choosing one initial point, we sample through the whole space of allowable outcomes. Then we probe each of these samples against observations, either cultivation curves, and as I mentioned before, omics data. And then based on this procedure, we stay with the set, in fact, with the population of models that are, in fact, statistically describing our problem. Now, the problem with this approach is that depending on the complexity of the approach, you might spend a lot of time of sampling the whole system and also in integrating data and trying to reconcile everything together. But whatever is the approach, once you end up with your dynamic models, they have to pass certain quality tests. I mean, it is like you're manufacturing a car. When you are selling your car and giving to your customer, you would like to have some quality a test that comes with the car. So we are also trying to make this together with our models. So once we create our models, we made sure that they have certain quality requirements. The first requirement is that they are always consistent with the quasi-steady state. This motivation comes from biotechnological applications where, in fact, when you're producing any biochemical of interest, the most of this biochemical is produced in an exponential phase. So you would like that your model really well explain this phase in a fermentation so that we can actually use it for the prediction. So if you look at this in our parametric space, it already gives you a certain range where your parameters should be so that you're consistent with this quasi-steady state. Now, once you build your models, they have also to be locally stable. Why we're asking for this? Because if you would do very small perturbation around your steady state, and if your model just explodes, it is not good for any kind of prediction. So for this, we put this as additional constraint, and it turns out that this really importantly, significantly reduces the allowable parametric space. Another requirement that we found in the very beginning, the most difficult to satisfy, was that, in fact, these are dynamic models, and they have to evolve with the time scales of biological systems. So we know metabolic processes are evolving in the scale of fractions of the seconds and mostly minutes. But if your models are not evolving the same dynamic scale, then they are not good for us, because we cannot effectively simulate what is behaving if we do any kind of perturbations in the system. And once again, as you see, this adds more constraints. And if you add some other requirements, you might have that your initial parametric space, which was very good, and you could find easily parameters, it turns out that your desired part in a parametric space, it just boils down to a couple of very small patches, which in fact pushes you to explore this space much more finely. You have to imply much more computational requirements to explore all these things. And here I just wanted to show, also to illustrate that it is not sufficient, even all these first three requirements that I had, they're not always sufficient. For example, on the upper panel on the right, what we did at the time, we built dynamic models of equali that were satisfying all these requirements. And if you see the yellow curves, they are replicating what was observed with the wild type. But then when we tested what is happening if we make genetic perturbations to our model and what was happening in reality, we see that our models, whereas they've been matching very accurately wild type phenotype, the recombinant phenotypes are just captured by a trend, but not actually very precisely. So what I'm saying is that if you would like to have models that are reliable for a wide range of applications, we have to add more and more data in order to make them of a higher quality. So it turned out that in this whole set of wild type models that were satisfying, there was a subset that satisfied both recombinant data. So you can find it, but once again, computationally, you have to explore much more in order to find. And this type of problems was, in fact, our main driver behind the research that we did the last couple of years, because we were asking, is it possible somehow, instead of trying to explore this whole region of the parameters, is it possible that we start to generate the models only within these green patches, so that we really reduce our computational requirements? And our first foray in this direction was a method that we called the iShrunk. And the idea, iShrunk like shrinking, like reducing the space. But the idea there was that we take, in fact, traditional kinetic modeling approaches like what we develop Oracle or Costas Maranas group K-FIT. You sample your space and information about the parameter values, and if these parameter values are providing certain property, in this case we were testing linear stability, we feed it to a CART algorithm. This is a classification and regression tree algorithm. And this algorithm, what it does, it in fact approximates this green space with the hyper rectangles, which is really nice because it gives you ranges on the individual parameters that are satisfying your desired behavior. So once you have these ranges, you just apply them once again in your traditional method, and you get, in fact, much better computational results. And we use this method, it works really well. I mean, we use it in a couple of publications to make responses to our models consistent with the multiple knockouts, to reduce uncertainty. And the main thing that we have seen in all these publications is that when you analyze these kind of systems, it is not all the parameters, in fact, that are really important that you know when you are trying to describe this behavior. It is just a few. In all these studies, it is like 6, 3 parameters. If you are capable of really precisely determining only a few parameters, you would be able to perform your task well. But whereas this works well for stability and consistency with multiple knockouts, the problem was that as soon as we started to pile up several requirements of these methods, it didn't work really well. I mean, it was mediocre. Like prediction accuracy was 60%, 50%, something like this. So because we understood that our space that we are trying to find, it's non-linear and we don't even know if it's contiguous. So we thought that a good idea in this case is to turn toward the neural networks, because neural networks, they are universal nonlinear approximators. And recently we got advent of all these generative machine learning methods where you are in fact trying to train your neural network so to create certain samples that are corresponding to your desired performances. So in this particular case, what we thought that it is a good idea to these samples that we were using previously for iShrunk, to use them to train our neural network. And based on this, we developed a method which is called Rekindle that we published, now 2 years ago. And Rekindle is, in fact, based on the concept of GANs, of generalized adversarial networks. And I don't know the background of audience here, but I will just go very shortly through it. So the idea of generative adversarial networks is that you have two neural networks. One, it is called generator. And the idea of the generator is to take as an input some random vector and to produce a vector of synthetic data. I mean, in ideal case, this synthetic data for us, it would be the parameters of our models. And there is, this network is coupled with another neural network, which is called discriminator. And the role of discriminator is to compare the synthetic data created by our generator and compare them with the training parameter set that comes from traditional methods. And based on this comparison, discriminator is supposed to tell you if the data that are synthetically generated true or false. And I've seen on some blog a really nice parallel to this. You can look at this like a generator is a forger, a guy who is trying to forge some masterpiece, and the discriminator is a curator. So the forger is providing forge the masterpiece, and the curator is supposed to, based on some other works of the same painter, for example, it is supposed to compare this and to make a decision if the painting in case is true or fake. And based on this information, based on the decision of discriminator, I mean the curator, we can use this information to improve the knowledge, the experience of the curator, but also we use this information to improve the knowledge of the forger, because if the forger knows that previous synthetic data set didn't pass the test, so he has to improve. So in this, if you see, it is in fact zero-sum game. One is trying to outcompete the other. And during the training of these networks, in fact, they improve performance each of them simultaneously, and then eventually at equilibrium, the generator is so good in reproducing the parameters of the training set that the curator, that the discriminator, is not anymore capable of distinguishing them. And once you have this generator, you can use it for your parameters. Now, in a particular case of Rekindle, we use conditional guns. The only difference with this concept that I was explaining is that next to the parameter data sets, you are also providing a label if this parameter set was belonging to this green patch or it is belonging in the space which is around. And once you have well trained your generator, then it really goes like a breeze. So you create any random vector, you put it in the input of your generator, and your generator, if well trained, is supposed to create you only the parameters within this green patch. And as an illustrative small case, what we did, we used this methodology to create models of E. coli central carbon metabolism. It is a small model. It is 64 ODEs. And we were supposed to identify 324 parameters, or around 300 parameters. And what I'm showing here is how the training goes in this particular case. So as you can see, as the training goes, that the percentage of the models that belong to what I call valid models, to a green patch, goes from something very close to 0 to 98%. And in some cases, we would go even to 100%. Now, once you train your model, you are asking yourself, is it possible that your neural network is generating models around 1 point? So essentially, you have one model, and this guy is just sampling from this very small space. So to check this, what we did, we used the Kalb-Leibler divergence measure, which in fact tells you information how close are the distributions of the parameters that your generator is producing and the training set. So ideally, they shouldn't be different. They should be like in this right upper panel. But when you start in your training with something like what we have in the right lower panel. And when we measure this thing, we see that, in fact, KL divergence really correlates well with our incidence of the models. That means that, indeed, our neural network is approaching in the distribution to original set. So we don't have that we're creating models from one singularity. Another test that we did here, it is also to observe what is the dynamic of the model. Because if I use a traditional method, Oracle, to create this type of models, I have a distribution that you see in a probability distribution function, which you see in blue here. And since our target was to have only the guys that are in the green patch, we were in fact biasing our generator towards having models with a faster dynamics. That's why the distribution is turned to the left. However, one thing here it is really problematic is that in order to get this type of neural networks, you have to have models already built. And depending on group, but some groups, according to what was reported, they need weeks and months to create a single model of such complexity. So the problem of generating good models for training, it's big. So what we thought is, okay, in this first approach is at least we know that for neural networks and in the image processing, there are methods that use transfer learning. So the idea is, can I maybe, if I have developed a model for one case, and if some other case comes, can I somehow use this generator for another case, but with much less parameters? And the idea is that when I train my generator for one physiology, it really gives me the parameters, but implicitly it also learns the structure of the network and also nonlinearity of underlying rate expression laws. So if I can take this generator, which already learned the structure of the network and nonlinearity, and use just a few data from some different physiology, then the hope is that my gun will just fine-tune the actual values of the parameters, whereas doesn't need to learn from scratch the structure and the nonlinearities. So we tried this in the same case study. When we integrated data, it turned out that we couldn't, based on all integrated data, there were still reactions that would operate either in one or the other direction. So if I would like really exhaustively analyze this system, I would need to create 4 families of models. So we said, okay, in the case that I've shown you already, we created one family of models. Can I use this generator to create models of the other families? And this is what we have done. And it turns out if I take a generator from what I call physiology 1 and I apply it to physiology 2, which differs from physiology 1 in a direction in one of the reactions in pentose phosphate pathway transadolase, we could only with the 10 models from the physiology 2 obtain a generator already with a 62% of good models. If we add more already with 100 models, we get plus than 95% of the models. In contrast, if you try to do physiology 2 from the scratch, before 500 models, you cannot do anything. I mean, these are all done with the 5 replicates, but we did many studies. It's really, it really transfers well. And then we did this from physiology 1 to physiology 2, 3, and 4, and all combinations. And in all cases, really, it allows you already with 10 models to get a good, to create the models for all other physiologies. And also we have tested what is this support of the distribution of our parameters, what are the responses, are the models robust, and it turned out it really worked well. Now, another thing that this type of approaches allows you, it is to analyze statistically your parametric space. In general, if you would start only with 10 models, you don't have really hard evidence that something is happening. But if you use 10 models to create your generator and then generate a population of the models that approximates these 10 parameters, and then to do analysis, then you get a much harder confidence in what is happening. So we analyzed, so we created our models and we analyzed the distributions of models which are valid and invalid. So you can in fact see that there are differences in the distribution of the models. Once again, we use KL divergence. And when you see the top parameter, we see that there is a slight bias toward the higher values of KMs in the case of this green patch population. So we took this parameter and we placed its values in 10 bins based on a distribution that we had, and then analyzed the dynamics of our system as we are walking from bin 1 till bin 10. And you can see there is a clear effect where as you are going toward the higher values of this parameter, that dynamic of the system is faster. So indeed, with this, we could identify which are the parameters behind the slower responses. And of course, for some other parameters, when you analyze this the KL divergence, you see there is no difference. And we did the same to see maybe it is not only about the distributions, but in fact, it turns out that many parameters are sloppy. Nothing, if you change their values, nothing is changing. So what was really nice in our opinion was that this method allowed us to create dynamic models very fast. As I was mentioning, in some cases, this creation takes weeks and months, but with this method, providing that you had some starting population of the models, you can train the models in up to what we see here, 1 hour. But once you have a nonlinear network, I mean a neural network generator, then you can in up to 20 seconds generate 1 million of models. So you can, it's really, really efficient. I mean, it is just filtering through a neural network. I mean, there is no surprise there. However, though this was really interesting, and I already mentioned what we could do with this, we were not happy because the problem is that, and everyone who was in this area knows, the problem is that just a few groups are capable of finding dynamic models of large scale. And if we would like to move the metabolic engineering, and I would say also synthetic biology community toward using this type of models and not steady state models, we need much more people to work there. And the problem with Rekindle was that you needed the starting population. So we thought, is it possible to train our networks without using previous models? And there, at the time when I was thinking about that, my idea was I had, for GANs, I had this generator and I had discriminator, but I was thinking why I would use now the discriminator when myself, I have already my system of ODs and I can use it to drive my neural network. Instead of having some curator, I already have a base fact. I have my system of ODs, which I can use to compute, in this particular case, linear approximation Jacobian of this OD system, and to use this as a measure to improve behavior of my system. And with this in mind, we turned to what is called now in machine learning world, natural evolutionary strategies. And the idea is very simple. So instead of having previous data set, you have a fishing net that you throw in your pond of kinetic parameters. And for each element of this fishing net, you evaluate how good is the behavior of your system. In this particular case, we are trying to obtain the models that have biologically relevant dynamics. So they are fast enough for biology, but not too fast. So for each of these elements, you estimate how good they are. And then based on this information, evolutionary strategy, in fact, computes a gradient in which I should move in this parametric space. And I throw another fishing net, which has hopefully improved performance. And then by throwing fishing net successively, we should hone in our desired range that we have here. And based on this, we develop another method, which is called the Renaissance. Yeah, we spent some time to thought how to make it, but it is based on this, and now hopefully soon will be published, but you can find it on bioRxiv. And how it works. So you start classically, as you start with any type of these methods where you are building the model. So you have to establish stoichiometry to put, do you have regulatory interactions, you take your rate laws, and then you do the data integration. So omics data. And once you have, to say, a scaffold of your model, then you use these natural evolution strategies where you create randomly in the beginning these generators. You use a random, some random Gaussian random vector to filter through this generator. You get your parameters. You estimate the dynamic responses of your system. Based on this, you know where to go, and you continue like this till you get hopefully where you would like to go. And we applied this to a bit bigger, not too big, approximately 90 ODE system, 90 equations. This is what we get. So already at 10 generations, we get more than approximately 70% of good models. And okay, we did the studies for different, but what was interesting for me is to see indeed how our training works well. So in the very beginning, we had the models that were either too slow or unstable, but as the training was progressing, you see how our distribution is moving exactly where we would like to go. So it really worked well. We also tested all our models for robustness. And for example, we would take the concentrations in the initial concentrations in these models. We would perturb them plus minus 50% and let the model evolve and to see does it going back to initial steady state. And we got all 100% of models coming back, meaning that the basin of attraction of these models was really big. And then once you have them, you can do, for example, here we used these models to reproduce the attenuate production in E. coli. These black points are experimental values, so you can do stuff like this. But another thing that you can do, you can use these models to reduce uncertainty in some steady states. And I will explain what I mean. I know it's a little bit. So what I would like to say it is, imagine that we have data like this. So someone gives us experimental data, and essentially the model that we would like to reproduce would have these responses shown by the yellow curve. So this is a perfect case. However, when you use steady state data, you might also reproduce the orange curve, which has exactly the same quasi-steady state, because the steady state doesn't know anything about this specific time. So the idea here is, instead of unnecessary reproducing models here, I will try to build the models only for the steady states that are not only have exactly the same values of glucose uptake and growth at the quasi-steady state, but also that have good, in this case, yellow dynamics. So if you do this, what we did, we took 5,000 steady states, and for each of these steady states, we created a population of kinetic models. We could do it because with the Renaissance, you can do in one generation of running is 1.2 minutes. So in 2 or 3 weeks, we could generate models for 5,000 steady states. So out of these 5,000 steady states, it turned out that something like 74% of the steady states allowed a good dynamics, whereas you could really trash remaining 26% of the models. So this allows you, in fact, to reduce the uncertainty also in the steady states. And yeah, I have only 8 seconds, so I will just go a little bit fast here. Another thing that you can go, it is that you can also not only discard certain steady states, but you can check in the individual components And based on their distribution, you can see that certain, when you compute variability of metabolite concentrations that are not measured, in this particular case, we had a range from approximately 1 till 3 micromole, 10 to the minus 3 micromoles. But it turned out that for all the values of 3-phosphoglyceric acid, for all values that are higher than 10 to the minus 3 micromoles, you cannot get good dynamics. So it is very likely that this intracellular concentration is somewhere in this range. And yeah, because I assume the organizers will. So I would just like to say that this work was funded by EU, Swiss and Swedish national science foundations and NVIDIA. And these are my collaborators, and we are all working together in the lab of Vasily Hatzimanikatis. Thank you very much.
Yeah, thank you very much for this very interesting presentation. Are there any questions here in the room?
I've got multiple questions, but I'll stick to one and probably talk to you after. When you were showing the potential different physiologies, you had like the graph for physiology 1, 2, 3, and 4. How do these different physiologies differ from each other? Like how different are they?
So this is they are differing in the directionality of reactions in TCA cycle and pentose phosphate pathway. So in one case, you might have that TCA cycle is operating in a cycle. In one case, you can have bifurcating cycle, for example. So in this particular case, I mean, I cannot say how big a difference was. That was our test case with the given data. And we did not test right now for something completely different. For example, starting from aerobic to anaerobic case. This we didn't test, and I cannot tell, but we didn't test it for the reason that we moved very fast to Renaissance, because Renaissance doesn't need training data, and it is much faster to work with it. But yeah.
Thank you. Yeah, thank you. I had several questions, but the first one is that it doesn't seem you have, at least in the model you showed an equation at the very beginning, it didn't seem that you had gene regulations taken into account. Is that right?
Yes, in fact, what we have allosteric regulations.
Yeah.
But gene regulations for this, this is in fact in our plan to connect with the ME models.
Okay.
Because to put for each enzyme, to put ODE to gene, I mean,
sure,
mRNA protein synthesis, and then how it affects.
But you had enzyme level, right?
Yes.
At some point in the,
Yes, I mean, right now we are estimating maximal velocities, which are enzyme level.
Okay.
But in the longer term, what we would like to see it is how to couple this with the regulation.
Okay. Yeah,
but right now we are not doing it because we are in fact stuck in the steady state part of ME models.
Okay, I see.
And then, we are. It is really on our planet.
So then, before you develop Renaissance, okay, you did all the software, and you needed to train the discriminator with real parameters. How do you get these real parameters? Are they really taken from actual experimental data, or?
We took 108 parameters from Brenda and Sabia RK database, these two databases, and remaining parameters we estimated.
Okay,
so
I see.
But that is a part of our method. It is that, as I said, you are not sure that even if they are measured, are they in vivo or in vivo? But the fact that they work together in one system that is consistent with all the data, it will bias some of them toward the system will bias them where they should go.
And the last question is, okay, all your systems, they can find out kinetic parameters, okay, they can put values, but could it also be used to actually go beyond and build a network? I mean, not from scratch, you know, but adding reactions and deleting reactions. I guess deleting reaction, you could set up parameters to 0, right? And yeah.
It can be used, I mean, we use it in a bit different context. It is more closer to RetroPath, where we have a network and we expand it by something.
Yeah.
And then if we do it like this, then yes, because if you would like to find a minimal network that you add,
Sure,
minimal network that you should add so that you reproduce the behavior, yes. But it's a really good idea.
Okay,
yes.
yeah, thank you, yeah.
So last one.
Hi, great presentation. Thank you. When you do the natural evolution method, you have some kind of fitness estimator. Have you tried using different fitness estimators and seeing sort of what set of characteristics biases your search more towards, like the sort of local minima or optimal space of your parameters?
I mean, yes, we have tested many. In this particular case, this was a starting point where we just wanted to have a good dynamics. But if you would like to represent this kind of behavior, because the fact that you have good dynamics means that only this slope here is good. But then a final titer of your models is not necessarily, it can be anywhere from here till twice or something like this. So when it really depends on what you are searching for, what are the properties you are going. But that's good with natural evolution, because you can create some ad hoc criteria and push your system. I don't know if this was particularly your question.
I was curious if you found any set of characteristics that drives the model towards better physiological representation.
This we did, but not through, we did in fact a posteriori analysis of what we have obtained. And I mean, I had it somewhere in the slides, but yeah, you can find some nice stuff.
Okay. Perfect.
Okay. Thank you.
Thank you. Thank you very much. So now we have a talk of Sutrik Das from the UCL, and he's going to talk about neural networks for engineered bacterial communities. So I'll leave the screen. Sutrik.
Good morning, everyone. I'm Sutrik. I'm from UCL, currently working as a postdoc. So I'll be presenting my work titled Designing Neural Network Computation across Engineering Bacterial Community. So I'll start with giving some basic background on neural network. So it's basically inspired by the biological architecture of human brain, and now it's a game-changing tool for a computation model. So it basically has nodes, which models as a neuron, and edges, which work as synapses. And each node experiences external information, which are signals, and the output of the nodes are basically computed by some nonlinear function, which is basically the summation of some inputs. So, and each node, which is termed as activation function, and the nodes and edges have the weights, which adjust itself as a learning process. It basically increases or decreases the input values. So now the artificial neural network underpins the field of deep learning with the help of machine learning techniques, has now doing a wonderful job in advancement of AI It can get it can generate complex function from data, and flexible to apply for various kind of problems like image classification, nonlinear regression, and everything. So inspired by the vast application of artificial neural network, synthetic biologists tried to get its own counterpart. So DNA and RNA being highly versatile and programmable in nature, it is now used for biological computation and showing exceptional usage. All this started with Tian's group around 2011 when they modeled neural network computation using DNA strand displacement reactions, which perform DNA computation and capable of recognizing pattern and decision making. Next, around 2018, the new thing came up as disease classifier, where around 2018, Lopez and group, they did some molecular computation strategy, which analyzes gene expression data to work as a disease classifier. They validated their model, which detects early stage cancer diagnostic, and also differentiate between viral and bacterial respiratory infections. One can look into this recent review article by Mia Moskin group, where they demonstrate the year-on-year progress of synthetic neural networks. You can see in the y-axis is the number of inputs they are using, which ranges from 1 to around 1,000. And most of them are worked on based on DNA, a protein, and they're also increased in the number of neural network sizes. With this background, I'll try to introduce my work, which is basically we want to do some spatial computation using mimicking neural network, which is basically microbes on a plate. So in our setup, these different colored of circles are basically engineered bacteria growing on an agar plate, and each bacteria is communicating itself with each other by a quantum sensing molecule, which are raising from one bacterial cell and basically works as a reader for another bacterial colonies. So it's basically a sender-receiver kind of network topology. And this signal reception and processing of this signal is now highly influenced by the location of the colonies, which is similar to the natural self-organization of bacteria to perform specialized tasks. Now the concentration is reaching the receiver colony can be tracked by this by approximate solution of this fixed equation of diffusion, where the first term becomes the input and the final term becomes the weight as comparable to the neural network. And in case of the activation functions, it's basically the bacterial response towards the diffusion molecule. So it is embedded in the bacterial population itself. So this is our complete setup where the bacterial population try to resemble artificial neural network on a plate. The biggest advantage of our model is that, as owing the addition nature of the input concentration, any arbitrary complex function can be performed by simply positioning the different colonies on an agar plate. We're starting with the method. So our goal is to optimize the network topology and try to spatially organize the colonies itself. So we are using something called genetic algorithm, which has already been discussed by previous speaker, which is based on natural selection. Iteratively, it modifies its population and try to get an optimal solution. So we start with the initial population where we have two layers, basically hidden layer and output layer, and try to add nodes with specific activation function embedded to it. We have two options here, is high pass and low pass. So with initial population, we assigned weights. Starting with, we are just defining weights as from 0 to 1, just to quantify the concentration properly, which is reaching to the receiver colony. Now we sort the population according to the fitness, and this is how we scan the top 50% and try to mutate the 50% population and to get some better results. Now to scan over all the possible combination, we use something called mutation operator, where the initial population, which has a definite combination of high pass and low passes, it goes under mutation under certain probability. It can go addition, where it can add a high pass or low pass. It can go deletion, where it can delete a high pass or no pass node, or it can just modify one of the node to high pass and low pass. And we have something called recombination operator, which just combine two different population, leading to a new population. So with this, we can scan over all the possible combination. Then we calculate the fitness, and within each generation iteratively, we guess the best possible topology where we can get what the activation functions are needed to be encoded in the different columns, and we can optimize. We can get the optimized weights also, which specially represent the spatial organization of the colony. So with result, we started with the most basic biocomputation, that is a logic gate. For an AND gate, we start with a hypothetical activation function where the range of the input value and the output value is same, just to as in a test case. So our desired output look like this, where the input values ranges from 0 to 50. With the genetic algorithm, we get this kind of topology where we have two nodes in the hidden layer and one in the output layer. All of the nodes have low pass, and we're getting this kind of output data where we can see the fold change is quite good. It's around 15. And we did similar thing with other logic gate functions, and we get pretty good result for OR, NOR, and NAND gate. We continued with our engineered activation functions, where you can see the ranges are quite different in case of high pass and low pass, but we're seeing better results with the same topologies in the hidden layer and the output layer. And though the fold change is quite different. Now to look into the spatial organization of the colony, without defining weights from an arbitrary range from 0 to 1, we define it as a colony, location of the colony, where we use a realistic range of diffusion coefficient and the time, and we varied the location from 0 to, let's say, 3000 into 10 to the minus 5 meter. And we did add some constraint to this model. We fixed the location of the output node and set a constraint that each node has to be at least 3 millimeter distant apart. We get pretty good result for all the logic gates and really good fold changes for the outputs. But this, I'll show you the spatial distribution. You can see this is for OR, NOR, and the NAND. You can just see this just by placing the location of the colonies differently, we can generate different logic gate function. And with this, I'll just summarize. So I'll try to mimic artificial neural network using microbial population, which are communicating via column sensing molecule. With genetic algorithm optimization, we get to refine the activation function which need to be encoded in the population. In the ideal case scenario, we're getting a significant high fold change, whereas when we have using the engineering activation function, but here we are just giving penalty to our network topology to minimize the number of nodes. So it gives a fairly good result, but with lesser fold change. With this, I'll just acknowledge. Thank you, my lab. Especially thank you to Kathleen and Ching they are doing the experimental part. Thank you for attention. Thank you.
Thank you very much, Chotrik, for your talk. Are there one question in the room?
Hey, as far as I understood, there are two parameters that you can change. One is the distance in X and Y coordinates, and the second one is the concentration. Am I right? Okay, then how you would apply genetic algorithm in these parameter set? Maybe I missed in terms of experimental setting.
Yeah. So in genetic algorithm, here we are just optimizing the weights. Here we are optimizing the weights, but in the when we're finding for location, we're optimizing the location itself. From the location, it calculates the weights, which is dependent on the time of the observation and also the diffusion, the diffusion constant of the diffusion molecules.
Okay, then how you translate this setting in the experimental scenario then?
So we have a plate reader, and we place the colonies in different locations. So it is already done, but I should have shown you some experimental result, but sorry, I don't have the slides with me. Yeah, so you just place different colonies around different positions, and the time we set for the observation, just drop down to that time and try to get to the output and try to generate this kind of logic gate function.
Okay, thank you.
Okay, thank you very much.